Good evening, everybody. Thanks for the introduction. So I'm just going to start by giving you a little bit of background into um, sorry, I'm just trying to make my that's worked. Into the ecology of the white clawed crayfish. So it's basically our only freshwater crustacean species, the only one native to England, Wales, and Ireland. We do have about 7 other species, but this is the only native one. Now, it's a slow-growing crustacean, and it matures fairly late, so it won't actually reach sexual maturity till about 3 or 4 years in the wild. In captivity, we can do that in the 2nd year, but more on that to follow. And it's basically, so it's slow-growing, and it's a long-lived crustacean, can live over 10 years, which is quite unusual. But it's also got this, it's relatively modest breeder, so the females only breed once a year, and they'll only produce up to about 40 to 50 hatchlings. So sometimes a little bit more, but usually around that average. But it's fairly flexible. It can live in both rivers and freshwater rivers and lakes, and it's an omnivore. So it starts off fairly predatory, eating plankton, and then it becomes more of a detritivore. And we call it both a keystone system, keystone species and an ecosystem engineer. It's really important for an environment. So if you take it out of an aquatic ecosystem, it can have negative impacts, and it's really important in terms of kind of being a detritivore and keeping down levels of sedimentation. So not quite so impactful potentially as a beaver, but very mighty in its own way. So in terms of its status, it's spread throughout Europe, but we are seen within in the UK, in England, Wales, and Ireland. We're still known as a stronghold for the species. And in Wales, as Muddy says, it's not actually in the River Dee, but it is in neighbouring catchments within Wales as well. Now, what are the main threats to it? The usual suspects, really. We've got water pollution. They're really susceptible to things like high nitrates, phosphates, chemical runoff, industrial pollution, agricultural pollution, but also, like many of our species, fragmentation, such as man-made barriers and reduction in waterways. But one of the most impactful threats to it are invasive crayfish species. As I said, there are basically 7 in the UK, but the one that's of particular significance is the signal crayfish. This is an American species that was brought into England in the 70s, mid-70s, and farmed very much like the American mink. It was farmed here. Ironically, we didn't really want to eat it, so we sort of exported it to our Scandinavian markets for food. But this crayfish is a much bigger animal, can live out of water for a few days, and so it basically escaped from the hatcheries and created havoc. So not only does it have this disease called crayfish plague, which in most cases can make a white claw population extinct in a few days, in a few weeks rather, but also it's a much bigger species, so it can outcompete our native one. So we set up a partnership in the Southwest in 2008, very much a lovely strategic partnership, as Maddie was talking about at the beginning, really getting the experts in. And we've got 5 key strategies, and we've had these going since 2008, but of all of it is backed up very much in research. We're a field conservation and science department in Bristol Zoo, so we're very hot on research, and it's really important to evaluate success of all of these elements. So I'm just going to give you a whistle-stop tour of kind of how we do this strategy to try and halt the decline of white clawed crayfish. So the first one is catchment mapping. So it's really important to know what you've got in your catchment. So with our focus of the Southwest, we really mapped the whole area. And you can see that before the crayfish before the signals sort of had spread, the white claws were kind of spread quite nicely throughout several of the counties in the Southwest. But by the time we started this project, the signals had spread throughout many of the counties, and the white clawed crayfish had become either extinct or massively reduced in their populations. So then that led on to ARC site establishments. So ARCs are a recognized conservation strategy for this species globally. And basically it's to move kind of threatened populations into safer sites. So these are sites that are discrete, they're isolated, they've got good water quality, and as for the foreseeable future, they're going to sustain a recruiting population fairly long time. So a long term, sorry. So how do we do these things? Well, we basically make sure that we adhere to the IUCN Conservation Translocation Guidelines. Now, these were written by the IUCN, and they have been basically devised for the Scottish. So for Scotland have made their own Scottish code. And I'm delighted to say that England has now followed suit, and we have got our own conservation translocation guides that was published by DEFRA in 2021. So we adhere very closely to this in terms of best practice and guidance to make sure that we are doing all of our conservation translocations, such as ARC site establishments, with these in mind. So how do we do them? It's a really complex process. We'll start doing desktops, really looking for potential ARC sites, safe sites within either river catchments or in still water. We'll do a site walkover to once we've kind of ticked some boxes, doing a lot of GIS mapping. And then basically it will start with the detailed surveys where we'll have lots of licenses and permits. Everything needs to be under both Natural England and the Environment Agency licenses. And we'll basically be looking to check for habitat, other species such as kind of predatory fish species, invertebrates, invasive species, and water quality to make sure that everything ticks its box. We also make sure that crayfish aren't present already, because we wouldn't move crayfish into an ARC site where they're already existing crayfish. So if everything is correct, and I would like to make the point that if we've got really endangered or high priority species in there, then we wouldn't be putting in white claw crayfish, because obviously we recognise that although it's an endangered species, we don't want to outcompete something that's also of priority or species concern. Now, quite often we'll do some habitat modifications along the way to make sure that we're kind of giving them what they like. Crayfish like little houses, so they like lots of refuges and lots of cryptic habitat to live within. And we'll also, where as possible, do some climate proofing. So that means if it's something like a still water site, then we might do some willow planting or some shading. And it may be that we do some more river profiling, really to create these nice backwaters that crayfish prefer. And then how do we do them? So with the donor stock, we try and make it sure that it's kind of within the same county, sort of as close a provenance as possible. Now, this could either be wild caught or captive bred, and it really depends on the scenario. So if you've got a really threatened population with signal crayfish kind of marching upstream towards them, then in that case, that scenario, it would be more, we would do a complete rescue of the animals we could. But if it's a case of where there's just a few crayfish left over or just hanging on in there, then captive breeding might be the more appropriate way for this. So with the translocation, basically, if it's wild caught, we'll be trapping them. If it's a captive born, we'll be bringing them from a hatchery. And then we'll acclimate them and we'll put them into kind of, appropriate little areas and pockets of refuges. And it might be a slow release where we actually put them into kind of refuges to give them a sort of helping hand. And the monitoring is super important. So this species can take years and years and years in terms of actually for us to find it. If you imagine a very large lake where you've put a few crayfish, it can take years to actually see them. They're very elusive, they're nocturnal, they're not that easy to find. So it's a waiting game. So we will do surveys, we'll do a walk over the first year we've released them, and then we will carry on. Basically, I'm still surveying sites a decade on. So then what have we done? So basically with ARC sites over the years, we've done 21 ARC sites over 6 counties, and that's a real mix of wild caught versus captive born. We've also done river supplementations, and this is a really nice strategy where you put crayfish above a native population that's maybe had a kind of reduced fragmented population where you can put them upstream so that you're really expanding the range to really try and build resilience in that wild population. And as I say, it takes a long time to find them. So just because some of them we haven't got a presence yet, then it doesn't mean that they're not there. And with environmental DNA techniques improving, we hope that's going to be a really useful technique for us in terms of monitoring long term. So moving swiftly on to the third strategy, that's our captive breeding. So that's in a case of where we basically breed them for where they've got, where wild caught isn't appropriate because they're very small numbers, but we still want to make sure that we're representing the population. And again, it's a recognised conservation strategy for this species, not just within the UK, but Europe-wide as well. So at Bristol Zoo Gardens, we've got basically these kind of upright hatcheries and then larger sort of larger vats as well. And we have 5 different biosecurity systems. At the moment, we've got currently 2 donor brood stocks, and we basically can produce about 1,000 hatchlings a year. We also do full cycle breeding. So we bring the females. This female on the right is a female in berry. She's with eggs. So we'll bring them into spring and let them release their hatchlings in the system, but we'll also mate animals and hold them and then basically rear the young as well. And we can achieve about a 90% survival, which in the wild, that would be more like a 5%. So it's a really good way of getting high numbers. And I also have them at home. I did my PhD on crayfish aquaculture, so I have my own little hatchery at home. And actually, we've got one in Wales, and Ollie's going to follow on to talk about the Welsh one. I did need to put that Welsh spin in as well. So he will talk about his hatchery in Brecons. So the fourth strategy is communication, really basically telling people about what they can do to help. And this is really talking about check, clean, dry. And I would urge you and encourage you to go to the non-native species website, the links here, just to sort of familiarize yourself, really talking about how we can stop the spread of plague and how we can actually stop, because plague is something where it can stay damp on your clothes and on your fishing gear, so you can inadvertently spread it from one river to another. So it's really telling you clean your boots, wash them off, and don't spread any invasive species along the way. And then the fifth one is a little bit more tricky. So this is our invasive crayfish control. So we're doing various different pilot studies, looking at a combination of factors. So integrated pest management, things like trapping, male sterilization, adding predatory fish, and also manipulating them at the gene level as well. So that's ongoing. So what's the bigger picture? Well, as Maddie said, a lot of these projects, the success of them is really looking at this sort of strategic partnership, really getting the experts together. So in the UK, we have this national UK crayfish steering group, which Ollie and I both sit on. And then we basically have this strategy where it sets targets. And then each region will have their own partnership where they deliver this national strategy, but basically relevant to their region. Arcsite and river supplementations are going on in both England and Wales, and Ollie will tell you a little bit more about Wales in a minute. And we're expanding crayfish hatcheries nationally as well. It's a recognised strategy for this programme. So we do offer expert advice and support. We run crayfish training courses. There's my little one up there for you if you're interested in joining. And of course, as I say, we back everything in research, making sure that what we're doing is good, making sure that we do adaptive management where appropriate. So I just want to finish by saying thank you very much for inviting me. Sorry, it's a little bit English, but hopefully Ollie will put on a lovely Welsh spin for you. And if you would like further information, another little bit of a plug, you by all means buy the book. And that's kind of the recognised manual from Natural England as well. Thank you.
Yeah, so Jen's given a comprehensive overview of what's been going on in the UK. I tend to focus on the work in Wales. We do a bit over the border as well in the marches, and I lead primarily on arcsite creation and captive rearing. Historically, depends very much on the geology of the distribution in Wales itself, but Upper Severn especially has always been pretty good. The Y is always been a stronghold for white clawed crayfish and the Usk. And on the Usk, actually, up until the mid 1900s, early 1900s, they still held a crayfish party annually at Christchurch College. So there was plenty about them. They were really abundant, and also in the southeastern valleys around Pontypool. Even sort of 2 decades ago, even a decade ago, there was still plenty of crayfish in the urban areas around these southeastern valleys. Wait a second, let me go back. Yeah, so now the very different story. Really now restricted to headwaters. They can still locally be abundant, but it's pretty bleak. We don't currently map the crayfish distribution in Wales. What we tend to do, we focus on catchments, then we'll extrapolate that data from that catchment to give us an overview, which isn't perfect, but it gives a pretty good idea, and it's not great at the moment. So what we're focusing on at the moment, as Jen alluded to, is arcsite creation. This isn't going to be our ultimate aim, but at least in the medium term, it should buy us some time. Big thing now with the gene drive, especially, it could be a possibility, and sort of inherent resistance to crayfish plague as well. But in the short to medium term, these arcsites should really give us a bit of breathing room almost. So you can see there in the bottom left, this is a buried female crayfish this was actually from one of the southeastern valleys, the tributary to the River Rumney. So she's got probably 40 or 50 eggs on there, and the female above her there, you can see the two-tone nature of those eggs. They're just about to hatch, and there is a juvenile there, just hatched. You can see the egg casing next to it. So this was when I was doing some artificial incubation trials during the 2007, 2008. We've tended to move away from that now. It's just maternal brooding. So I'll take these females in from the wild in April and May. I'll take those back to the hatchery. We'll set up refuges for them, feed them. The eggs will hatch normally in June, and the female will continue to maternally brood them sort of post-hatch. That juvenile in the picture there, that hasn't actually got any mouth parts. It's still living off its yolk sac. So they'll undergo about 3 molts, then they become relatively self-sufficient. I'll take the females out of the tank and return those back to point of capture. So at the moment, we're running 4 research systems. This is the most recent one up the top there. That is, when did I create that? Probably about 3 years ago. Use fully recirculated water, primarily for biosecurity reasons. You can imagine if we were obstructing water from our adjacent stream, all it would take was an otter to change catchments upstream, an angler, a canoeist who'd recently been in water carrying crayfish plague, and that'd just be game over for us. So again, all these systems are discrete, so we can use them to rear crayfish from different catchments, different populations. Initially, I was using sort of semi-natural rearing techniques. I'd put gravel, other refuges in the tanks as it was then. Tended to now move towards these troughs, these Canadian-style troughs with the synthetic substrate. So you can see in the bottom right, that's a biobun, salmonid hatching substrate. The alvins would normally take refuge in this, and it works really well for juvenile crayfish as well. You can see them sort of holding around that structure there. The beauty of the research system is we can manipulate temperature, particularly photoperiod. So if I want to breed crayfish in our captive environment, you can drop the temperature right down. At the moment, these are running at about 10.7 degrees. So this is almost mimicking the sort of natural temperatures, just to give us a reasonable growth, but I don't want to go too high. I'm pushing on a bit now. Yes. Right. So this should give you an idea of what it looks like in the tanks. That's the biobun being lifted up there. And these would have hatched for probably 2-3 weeks, maybe a bit longer prior to this video being taken. It's really useful. You can just take up all this synthetic substrate and essentially just scoop the crayfish out. So it's really useful. That system on the previous slide, I can probably hold 1,300 to 1,500 crayfish in there. So you've got those on tap, ready to go. Whereas if you were taken from a wild environment, you'd have to either do drain downs, trapping. It just makes things that much easier. And we know these crayfish are disease free as well, so we can have confidence in that. Just to add something Jen said about the IUCN stuff. We are actually, for 3 years ago, we started adopting the IUCN conservation translocation stuff. So we're actually following that now, which is useful, just to give us that bit of structure around any moves. So to date, released nearly 6,500 captive reared crayfish. It seems to work pretty well. I don't think there's been a site yet which I haven't revisited and not found our captive reared crayfish. We're also starting to get first generation wild recruitment. This is actually a site on the upper Hondy, a tributary upper Hondy, which is a part of the mono on the left there. You see these insulated boxes. This was a site stocked in autumn 2021. So we've got 6 arc sites on the go at the moment, and more in the offing. This is that site. This is in the Mono Valley. You can see that's the notebook release site there, and this is the Escaley here. This is our donor site. Interestingly, this was we used to be able to collect from the whole bottom of the Escaley here right up to the top, but around about sometime between 2014 and 2016, we lost everything from Michael Church Escaley downstream, well 90%. I think that was a pollution incident at the time because we were still picking up the odd crayfish below that. But if you think about the biomass, talking about the knock-on impacts for the rest of the ecosystem, under a good sized stone there, you could pick up maybe 4, 5, 6 crayfish. So you're talking hundreds, if not thousands of kilos of crayfish lost from that ecosystem. Since then, we've managed to find crayfish above Michael Church Escaley, probably a kilometre, 2 kilometres above that. Then sometime between 2020 and 2022, we lost those as well. Apologies. And now they're just left this very sort of remnant top end there. This is the only place I could find crayfish in 2022. And this arc site here, this is you can see the waterfalls. We're talking about sort of discrete biosecure areas. This is the first waterfall, maybe 8 to 10 feet high, and this is the next one above it. Didn't get too close, but you've probably got 30 to 40 feet drop from. So we know we're not going to get any signal invasion up this stream in particular. This is releasing some crayfish on the site. These are aged 2+. So these should have bred this previous winter. So interestingly, when we start this survey season, we can see what's going on. Whether we've had any wild recruitment. So yeah, we're continuing to rear for the notebook and the mono. I went back this year during the dry weather, and I found that we still had crayfish there. So that's 12 months on. I turned over 10 stones and found 3 crayfish. So that was pretty encouraging. Beginning to work further north, so we're looking at the upper 7 as well. That's been really good. Been working with Montgomeryshire Wildlife Trust. So they've been doing some useful eDNA stuff. You can see there, I think this is a mix of Severn Rivers Trust data from 2018 and Montgomeryshire stuff from 2021. And we're looking at this area around the Banwy. Seems to be a relatively stronghold there. We did some preliminary survey work this summer, found some good numbers there. So that's probably going to be a reasonable donor site. Then we're just trying to narrow down potential arc sites at the moment. This was the big thing from this year. I found everyone knows how dry it was, but this was actually one of the donor sites I've been using. This was full of water and full of crayfish back in April 2022. This was taken in August, and this was dry for 6 weeks. So what was a stronghold for the catchment itself should be less so now, and it's not something which is in isolation. You think how dry it was in 2020, 2018 as well. So if this continues, it's going to have severe implications for the species across Wales and across the UK and wider as well. And yeah, it's always worth remembering if you can just get into the habit of cleaning your kit off and spraying down. You can get Virkon Aquatic online, iodine-based disinfectants. Virkon's probably the best. It's relatively inexpensive. You can use bleach. You can use really hot water. But if you can just make a habit of, if you know you're going to be moving around the catchment, just to make sure you clean all your kit off and you do disinfect. If you can't disinfect, just dry it out thoroughly. As Jen said, the plague spores, they can last for at least a fortnight in damp conditions. So it's really worth making the effort.